There you go. Nice. Great. Well, it is my honor and pleasure to introduce Santiago Demajo, who is a senior postdoctoral researcher in the biomedical genomics lab and the scientific coordinator for CGI-Clinics at the Institute for Research in Biomedicine, or IRB, in Barcelona, Spain. So thank you for joining, Santi, Olivia, Nuria, all from Barcelona after hours at 5 p.m. Barcelona time. So we appreciate you taking the time to be here and looking forward to learn what you've been up to.
Perfect. Thank you so much. I will share my screen. Okay. Do you see it correctly?
Yep.
Okay.
Yes.
Perfect. So thank you again for the invitation. It's a pleasure to be here and share with you our work and our project regarding CGI-Clinics, and specifically I will focus mainly in the part of the project about the identification of cancer driver mutations to improve genomic interpretation. As all of you know what precision oncology is, of course, so the Cancer Genome Interpreter Clinics project is focused in improving interpretation in precision oncology. As you know, when we sequence the sample from a cancer patient, we get a list of genomic alterations. And before taking decisions regarding the treatment or in general the management of the patient, a key step in the process is the interpretation of cancer genome variants. So when we wrote the project about 2 years ago, we identified 3 important challenges in the field of precision cancer medicine. On one hand, as you know, although this has been improving in the last years, the interpretation step is not systematized. It is done differently in different countries, in different hospitals, and even between different people in the same institute. Another problem is that even if you do the interpretation very well, the best way you can, there's always variants of unknown significance. So actually, a high proportion, the majority actually of mutations observed in cancer patients are variants of unknown significance. And then there's a third challenge that I will not touch today, that is regarding the patients, that all this information, of course, is very complex for patients. So within the CGI-Clinics project, we propose some solutions to tackle these challenges. On one hand, we have a tool called, that some of you may know, called Cancer Genome Interpreter, CGI. So we are improving this tool to advance in this systematic interpretation. Then for the variants of uncertain significance, we propose the automatic learning to try to interpret these variants. And finally, we have the third leg regarding patients. Just to put a bit more of context, just say that the challenge in variant interpretation is expected to grow in the future. As you know, sequencing costs are dropping, and also in line with this, whole exome and even whole genome sequencing are arriving to the clinics. So these technologies generate large amounts of data, and of course this leads to an increased challenge. So the problem of interpretation is important now, but it's expected to become bigger in the future, and therefore tools like CGI are important now, but even more essential in the future. Okay, after this brief contextualization, just to mention that I will divide my presentation in 2 parts. First, I will talk about the Cancer Genome Interpreter tool and in general the project, and then in the second part I will focus in the automatic learning to interpret variants of uncertain significance. So as some of you may know, the CGI tool that we already actually developed some years ago in around 2017. It's a tool that it's online, so you can enter to this link and use it. And basically what it does is to interpret the mutations from a patient, from a cancer patient, and gives mainly two main results. On one hand, it identifies the oncogenic or driver mutations, and on the other hand, it identifies potential biomarkers linked to these mutations. So for the first part, the identification of oncogenic mutations, CGI uses basically two blocks of data. On one hand, it uses databases, both internal CGI databases and external databases like ClinVar. And on the other hand, it uses machine learning and computational algorithms. So taking all this together, each mutation, this is an example of a lung cancer patient, is classified in oncogenic or non-oncogenic. And regarding the second part, the identification of biomarkers of drug response, as you all know, the biomarkers are characterized by a specific alteration linked with a drug treatment in a specific tumor type, in this case, lung cancer. The mutation can give rise to responsiveness or resistance to the drug. Biomarkers are classified by level of evidence in CGI from A up to D. A means, as most of you know, clinical guidelines, and we have VICC for the clinical trials, and D. And also the source of this evidence. So this is basically the tool that we have now, and this you can find online. But now the main objective of the CGI-Clinics project is to develop an enhanced version of the CGI tool. This is what we are already doing. That meets the needs in the clinics, and then the final objective is that this tool can be implemented in clinical practice. So to achieve this quite ambitious, I would say, objective, we have a great team of 15 European partners. As in IRB Barcelona, we are in charge of on the technical development of the tool. We have an important collaboration. So we have within the consortium, we have up to 10 different hospitals from 5 European countries that are involved in the project. We also have the sequencing center here in Spain, CENAC, also patient associations and some other partners involved in communication and dissemination, and also in the regulatory and real-world evidence part of the project. This is a 5-year project that we started 1.5 years ago, more or less, and we'll finish in 2027. As I was mentioning, one key part of the project is that we have 10 European hospitals from 5 countries, Spain, France, Germany, UK, and Greece. And the idea is that the clinical community is directly involved in developing and testing the tool to be sure that the tool addresses clinical practice needs. We are also establishing collaborators with other hospitals. And importantly, in the last part of the project, in the last 2 years, more or less, we plan to extend it to 30 hospitals that will be able to test and improve the CGI tool. I cannot see you, so if anyone has any questions or comments, just talk, because I don't see your faces. Okay, and also part of what I wanted to share about the project per se is the main values that we have. We are a community-driven project. We want to follow the principles of open science, and our main objective, of course, is the benefit for patients. And this involves several things. For example, we are a collaborative project. We like to collaborate with other initiatives and projects and institutions. For us, the public interest is the main goal, to build something that can have a public impact. Regarding open science, for us, transparency is very important. We don't want anything to be black box, as I will also mention regarding the algorithms. And we want to facilitate data sharing. And regarding the benefit for patients, we want this tool to be used in the clinic so that patients can benefit. We also would like to promote equity and importantly cover needs that are currently unmet in the clinics. Okay, so this is coming back to the Cancer Genome Interpreter tool. This is a schematic representation of how the tool works. Basically, you input a file or a list of tumor mutations and the tumor type from the patient. And the pipeline of the analysis takes in consideration both expert-curated databases and what we call the automatic learning platform to identify the oncogenic mutations and the biomarkers of drug response. And this is a representation of the tool we are building, the new CGI tool, which will have improvements in all the different parts, input, output, and analysis of the tool. We will also have a new user interface that should be intuitive and easy to use, a new reporting system, easy to understand, and with different layers of reports to tackle specific users. We'll have two additional modules, one regarding virtual molecular tumor boards. So when the automatic interpretation from the tool is not sufficient, there will be an option to ask for expert support with this module. And also we will have a module regarding data exploration, so statistics of all the aggregated data introduced in the system. So the idea is that we aim to have the first version of this new tool, this new CGI tool, that would like it to be for comprehensive and intuitive cancer genome interpretation in approximately 1 year. In about April 2025, we expect to have the first version. Then there will be some other iterations, and we'll have an even more advanced version at the end of the project in around 2027. Okay. Now I will switch to the second part of the talk regarding focusing on the automatic learning platform used by CGI. Basically, when we talk about automatic learning platform, we refer to several computational methods and machine learning based algorithms that we have inside CGI that are involved in the interpretation of mutations. And I will talk about two of the two most important ones, actually, that are called Intogen and BoostDM. And basically, this question, the question of which are the genes and mutations that drive tumorigenesis, has been one of the main questions of the lab, I would say from the very beginning of the lab. And we have developed during the years these two approaches to identify which are cancer genes with Intogen, and more recently to identify cancer driver mutations with BoostDM. So I will now discuss these two approaches. Going back to starting, from the cancer genes, as you know, there's an extensive literature about cancer genes. We know a lot about a lot of cancer genes, the relevance in specific tumor types, their mechanism of action. So we really know a lot. There are for some other genes that are less studied, maybe we know less, but there's a lot of literature. But we wanted to approach this problem from a different perspective, like to try to build a comprehensive way of identifying all cancer genes in each tumor type using computational approaches. So The idea here is if we can identify cancer genes by learning directly from the somatic mutations of thousands of tumors, and this is what our methods are based in. And more specifically, an important assumption in both for Rintogen and BoostDM is that tumor development follows Darwinian evolution. As you know, our cells get mutations during life, and this is the variation. Most of these mutations will have no consequences for the organism, but some of them may provide selective advantage to the cell. This is what we define as driver mutation. And this will lead to the selection of this cell, of this cell with this driver mutation, leading to clonal expansion and eventually to the formation of a tumor or to the appearance of cancer. So our lab and others have shown in the last years that identifying signals of positive selection is an effective way to find cancer driver genes. And this is what we do in Intogen. The idea of Intogen is to analyze the mutational pattern of the genes mutated in a specific cohort of patients. For example, we do the analysis tumor type by tumor type. We take a cohort of patients from a specific tumor type, and we analyze all the genes mutated in that tumor type. And what we try to find is these signals of positive selections that can be classified in three main groups. Recurrence means, as you know, that when we observe in a specific gene, that this gene is much more recurrently mutated than we would expect by chance. This is a clear indication of positive selection. Also, as you know, in many genes, for example, in oncogenes, mutations are placed in a specific clusters, not randomly distributed but located in specific clusters or even hotspots. This is another signal of positive selection. And finally, the functional impact. When we see that most of or high proportion of mutations compared to what we expect by chance are functional mutations like means, missense, or nonsense, for example, this is also a high indication of positive selection. So the Intogen pipeline basically puts together 7 computational methods that identify the signals of positive selection, and using this, we identify the cancer drivers. So this is data from our last release of the method from last year. We have been putting together during the years different cohorts of cancer patients, whole exome and whole genome sequencing. And in the last release of the method, we have 33,000 tumors for 73 cancer types. So in each of these cancer types, what we do is we analyze the mutational pattern of all the genes that are mutated, looking for these signals of positive selection. And at the end, we arrive to this final picture. This is in our last release. We identified 619 cancer genes across 73 tumor types. So with this approach, we are able to recover most of the well-known cancer genes, but we also have the possibility to identify new ones and also identify new tumor cancer gene, cancer type combinations. So we know in each cancer gene which are the drivers. You have all these results in the Intogen website. Here is a review that actually talks about our previous release, but still it explains the method of Intogen. So this was like the full, the summarized picture, let's say, of the Intogen output, but here we have actually the results that are tumor type specific. So for each tumor type, and here we have 4 examples, lung, breast cancer, AML, and melanoma, we have a list of the genes that have signals of positive selections and therefore are the drivers of each tumor type. Okay, so with this, we believe that we have a quite complete picture, I would say, of all or most of the cancer genes, especially or specifically in the tumor types for which we have enough data. Of course, for rare cancers for which we don't have data, we cannot use this approach. But for the common cancers, I would say that we have a quite complete picture of which are the cancer genes. But then we have the second problem. If we analyze, for example, these around 30,000 tumors from these different initiatives, and we analyze exclusively the cancer genes in these cancer patients, and we go to databases like LIMBAR or OncoKB, what we see is that most of these mutations are not annotated. I have to say that this plot is a bit outdated. We are updating it, but the conclusion is the same. I mean, this blue bar goes a bit up, but the conclusion that most mutations in cancer genes are variants of arithmetic significance is still completely true. So how can we know which specific mutations in cancer genes are the ones that drive tumorigenesis? So maybe the first approach that one would think about is an experimental approach. Of course, one thing that we can do, and this has been done and is being done by different groups and different initiatives, is what can be called saturation mutagenesis. So basically, you mutate all the possible residues in a specific gene. You introduce it in an experimental system, a mouse model or a cell model, and you study a specific phenotype. This, of course, is very interesting, and as I was saying, it has been done for important genes like TP53, but of course it has quite some limitations, both in terms of cost and also that at the end you are using an experimental model. No, the ideal experiment would be, and of course nobody will do that, and nobody wants to do that, but the ideal experiment would be to introduce a mutation in an individual and to see whether it creates a tumor. This cannot be done, but actually we can say that somehow we already have like a natural experiment. If we put together the genomic data from cancer patients, we already see the mutations that have occurred and have been selected through the evolution of these patients. So the approach that we follow here is to use machine learning from this human data in an approach that we call in silico saturation mutagenesis. So the idea is the same. Can we identify the cancer-driven mutations by learning directly from the somatic mutations from tumors from patients? I don't have time to enter in detail in the methodology, but we can discuss it further if you are interested. But this is the summary, right? We developed this method called VICC. This is published, so you can read more about this in this paper. Basically, VICC is a machine learning-based method for in silico saturation mutagenesis that what it does is to learn the combination of features that define driver mutations in a gene and tumor type specific manner. So we start from this cohort of around 30,000 tumors, and we have been able in the last release, because now we are working in a new release, but in the original release of the paper, we were able to build 185 predictive models. These models are gene and tissue specific. And importantly, as I will show you afterwards, they are interpretable. So we don't want this to be black boxes that you get a prediction, but you don't understand it. But we want to understand all the predictions. So this is an example of one of these models. this is, for example, TP53 in colorectal cancer. What you see here is the sequence of TP53, and what we show here is this in silico saturation mutagenesis. So basically, we in silico mutate all the possible we simulate all the possible mutations that could occur in the gene. For example, if the first nucleotide is an A, we simulate the mutation to C, T, and G. And for each mutation, we predict whether this is a driver or a passenger. And this is what you see here, this Boost DM score that goes from 0, that means passenger, to 1, that means driver. And we have for any possible mutation the prediction. So we can see here, for example, that most of the drivers are concentrated in this P53 domain of the gene. And in the bottom part of the figure, what you see is really the characteristics, the features that are used to do this prediction. So each model uses some of the characteristics that I showed you before in Intogen that are signals of positive selection. So for example, if the mutation is in a 3D cluster, in a linear cluster, if the mutation is in an important domain, if it's conserved, the type of mutation, etc. So basically the idea is that Boost DM classifies all potential SNVs as driver or passengers, and therefore gives an annotation to all variants, including the Boost. Again, all this information is publicly available here in this link in the Boost DM webpage.
So, Santi, I have a question.
Yeah, sure.
There was a question that came up in the chat about the use of MAVES.
The use of?
Do you have any plans to incorporate MAVE data into your models? So, like, I don't know, Wes, do you want to elaborate at all?
No, in principle, no. So we use the MAVE or the experimental data. We use them to compare it. I mean, I will show you afterwards that we have used some experimental saturation mutagenesis to compare the performance of our approach versus experimental. But in principle, we don't plan to include MAVE data in the learning. So the idea of the learning is that we want to learn directly from data in patients. So we don't want to be learning from an experimental model that, as I was mentioning, I'm not saying that it's not important and it's not interesting. Of course, it's very interesting, and those are really important initiatives, and we of course take this into account to compare and to validate our method, but we don't plan to use it like to learn from.
That's good to know. So when you were using it to validate against the experimental, did you find, I'm assuming that you found like really good concordance between the computational method and the experimental method for determining drivers and passengers?
Yeah, I will go to this in 2 slides, but yeah, the idea, another thing that is important for us is to mention is that at the end, in MAVE, you are using an experimental model. For example, for TP53, there has been, if I recall correctly, 4 or 5 different saturation mutagenesis experiments or MAVE, and they don't give exactly the same result. So for us, we consider this as a relevant information, but not the ground truth. So of course, we see a lot of coincidence, but we don't consider the results from MAVE experiments as the ground truth, but as an important factor to have into account for comparing. Okay, so I will continue, and I will arrive in 2 or 3 slides to this precisely to this comparison with experimental data. I was going to mention here that this is another example. This is another gene, NRAS in AML. As you see here, the model is completely different. We only predict as drivers very few mutations, and all the rest are predicted as passenger mutations. Of course, this is an oncogene. It's very different from TP53, so it makes sense. And here you can see a third example. In this case, is EGFR in lung adenocarcinoma. And here I would like to talk about this thing that is very important for us, that is that the models are interpretable. And I will explain this with an example. So for each of these dots, for each of these predictions, we have an explanation linked to this prediction. For example, I will put the example of this important mutation that if you are working with lung cancer, probably you will know that this is a very relevant mutation in lung cancer in position 858. So this is predicted as driver with a VICC score of 1, so a very reliable driver. And for each, in this case, we have Y, no? We have the explanation of Y. In this case, this mutation is predicted as driver because it's in a 3D cluster observed in cancer patients, also in a linear cluster of mutations in cancer. It's in an important domain, in a tyrosine kinase domain. It's a missense mutation and it's in a residue conserved through evolution. So all these elements together leads the model to predict this as a driver mutation. And basically the idea is that for each mutation, for each prediction, we have something like this. And actually, I would like to mention that we are working in ways of showing this in a more, let's say, user-friendly way, so that you see the important results easily, thinking especially about clinical users and users of the Cancer Genome Interpreter. But the idea is that we have the explanation for each prediction. And I come back here to the point of experimental saturation. So this is, of course, an important part of the article, and we basically show that VICC outperforms experimental saturation mutagenesis. This is a comparison between VICC and two of the TP53 experimental approaches. Actually, at that moment, there were only two. Now, I think there are 4 different studies that we are also including in this validation. And also in the paper, we show that VICC also outperforms other computational approaches in identifying driver mutations. So coming back to this plot I showed you before, if we annotate the same collection of mutations with VICC, what we see is that we are able to annotate up to 50% of the mutations observed in cancer genes in this cohort of around 30,000 cancer patients. So, of course, this is an improvement respect to what we have in the databases, meaning that we are able to predict the oncogenicity of mutations that are variants of uncertain significance otherwise. As I was mentioning, we are working in a new release of VICC, so we expect to have new models for more genes soon. So we will update all this information. So the idea is that, coming back to CGI, we use these computational approaches, Intogen, VICC, and others, inside CGI as part of the interpretation, that in the part of classifying the mutations in oncogenic and non-oncogenic. Okay, now I would like to talk about an internal discussion that we are having currently in the lab, and we believe it was interesting to share it with you. that is about the relevance of computational methods in the classification of somatic variant oncogenicity. That is very related with CGI, VICC, etc. So I don't need to introduce this because you are basically part of the authors of these guidelines. As you know, this is the guideline for classifying pathogenicity of somatic variants in cancer. The classification is based in a scoring method, and at the end, variants are classified in 5 categories from benign to oncogenic. And to do this classification, different data is used that can be divided in basically these 5 categories. The first thing I want to mention here is that, of course, we believe that systematization is super important, especially in the clinics. So all this effort in systematizing interpretation of somatic variants is crucial. And actually, we plan in CGI to progressively align with some parts of these guidelines. For example, we will include things that we don't have currently for the new version of CGI, like population data and cancer hotspots data. But one thing that we wanted to discuss with you, and that we are also discussing within the CGI-Clinics consortium, is the importance, we believe, to consider if computational methods may have highest importance in the future. As you know now, the computational evidence only gives one plus or minus point compared, for example, with experimental data that gives 4 points, for example. One thing that we would like to highlight is that, of course, methods are not all the same. The scope of the methods is computational methods, similarly to experimental methods, is different. In experimental methods, of course, it's not the same an experiment in a cell line that's something much more sophisticated, like I don't know, a patient-derived organoid or a patient-derived xenograph experiment in mouse. So we believe that in computational methods, also the scope of the different methods are different. And focusing again in VICC, for example, as I explained, VICC is based in learning directly from the mutations of cancer patients, and actually takes into account many of the features that are already included in the guidelines, like, of course, for the predictions, the consequence of the mutation as nonsense, missense, etc., is taken into account. In this case, in a specific manner, not only considering if it's a tumor suppressor or oncogene, but the specificity of each gene. Also, the presence of the mutation in functional domains, it's also taken into account. And regarding the cancer hotspots, we don't take into account the hotspot, but something that we believe is a bit more broader, that is considering clusters of mutations observed in cancer patients, both in a linear or in a 3D way. So at the end, what VICC is basically doing is analyzing the combination of these features and others, such as conservation, in a gene-specific manner to do this annotation of mutations. So one part in which we believe that in the future methods like VICC could have an impact is in the classification of variants of a certain significance. That at the end is variants that are classified, but at the end they are classified with no classification. So basically, we don't have enough evidence to classify them as oncogenic or benign. So we are exploring how VICC may help here in the notation of variants of a certain significance. This is something ongoing that we are exploring, and we are studying together with the clinical partners. And I wanted to share with you just one example that we have seen in the collaboration with the hospitals. That is some examples of missense variants in cancer genes observed in a patient that hospitals annotate them as variants of a certain significance, and CGI, and specifically VICC, classifies them as drivers. And here is a particular example of a missense mutation in a TP53 classified as VUS in the corresponding hospital and predicted as driver. And this is the explanation of why this is predicted as driver. This mutation is present in a 3D cluster and a 2D cluster of mutations of certain patient, is in a conserved residue, and is a missense mutation. So altogether, this leads to the classification to the prediction of driver. So the impact of this that all this could have in the clinic is something that we are very interested in, and we are exploring together with our clinical partners. And we can, if you want, we can discuss it a bit at the end. Actually, I'm arriving already to the end of the presentation. And I would like to go back to this figure and now focus not in the blue part, but in the gray part. So with Busiem, we are able to classify a lot of mutations, but there's the other 50% that we cannot classify. And this is because we don't have enough data to build models for all tumor types, all genes in all tumor types. So another important part of the project of the CGI-Clinics project is to have access to more data more genomic data that currently is produced mainly in the clinics. So we believe that accessing more data, we will be able to build more and better predictive models. And the basic idea is that if CGI is used in the clinics to interpret the mutations, and this is useful, for this interpretation, as I was explaining, one part of this interpretation relies in this automatic learning platform. But if this data is shared internally in the system and we can keep learning from the new data, this of course will lead to improved models, improved predictions, and at the end it will lead to improved interpretation for future patients. So the idea is that by using CGI, patient's data is one click away, let's say, to be shared, of course, in a secure way, and of course with all the important aspects to take into account for the benefit of future patients. So with this, I arrived to 4 basic take-home messages of what I explained you today. First, the CGI-Clinics project aims at developing a comprehensive and intuitive CGI tool for cancer genome interpretation. And the idea is that we want this to be adapted to the clinics so it can be implemented in clinical practice. The automatic learning platform is a key component of the tool. And the idea is that this can be helpful to predict oncogenicity of mutations, including variants of percentage significance. Third, I didn't enter in detail in this, but we aim to test the clinical performance of CGI, including these predictions, the automatic learning platform, and compare it to standard of care. And finally, patient data sharing is essential to improve the automatic learning and therefore being able to improve interpretation for future patients. And with this, I would like just to thank all the people from the Barcelona Biomedical Genomics Lab, that, as you know, is led by Nuria López-Vigas, all the people from the CGI-Clinics Consortium. This is a picture from the kickoff meeting that we had 1 year and a half ago, more or less. Of course, thank you to patients and families and all the funding agencies. And thank you so much for the attention, and I will be super happy to discuss this further. I see that there are maybe more questions. I will stop sharing. Maybe.
Yep, we can open up for questions. Thank you so much. That was really some great work and a lot of information. So thank you so much. Not sure who raised their hand first. Maybe Brendan, go for it. Okay, Wes, go for it.
Okay, great presentation, Santi. Excellent. I think in terms of what you mentioned in trying to actually increase the score that could be applied with like Boost DM within the actual oncogenicity framework, I think that's something that we could absolutely do and follow a similar method that's what's being done with MAVES and the ACMG guidelines right now, where you basically go in and you take variants that have some ground truth, right? And you apply, this experimental model on top, right? And you basically see like, okay, does your boost DM classify, the truth the same way that you would actually go through and do that? And then you can apply that across. I can send you the preprint that the MAVE group, along with some of the people who've been working on ACMG, have done. So they've actually done reclassification for like VUS variants within BRCA, and P10 and TP53. And so I think we could follow a similar model, because if the data correlates with that ground truth, they're actually making it so that I think within the next version of the ACMG guidelines, you'll actually be able to apply an upgraded classification and strength to that, using that tool. And so I think this is something that would be really awesome, as you're kind of able to go about and validate. And the way that they do that is per gene right now, where you kind of have to do that. But I think that would be a really wonderful resource for being able to, classify some of those VUS and some of those genes, and push those either into likely benign or to likely oncogenic.
Sure. Yeah, this could be super interesting. So we would be super happy to collaborate with you in doing this validation. Actually, we are planning some things that in the same line together with the hospitals. Currently, what we are doing basically is to have this dual classification. On one hand, we have the as the example I showed you, the classification from the hospitals, and then we compare it with the classification of CGI. And this allow us to do like two main comparisons. On one hand is like let's say the validation of CGI. So variants that are clearly classified by the hospitals in pathogenic or in oncogenic or benign. We can study the performance of CGI here. And also regarding the variance percentage significant, right? To as example I was mentioning, to learn what is CGI and boost DM telling us in variance percentage significant. So we will work in these two lines, but of course we would be super happy to also do this approach like the MAVES.
Yeah, and for instance, like what that group did is they worked with Ambry Genetics so that they had a larger N, right? So they could actually get the amount of data that they needed. And then Ambry actually went back and reclassified variants for patients who had been seen using their panels. And so it's a nice kind of feedback where you can provide patients with updated results that are more meaningful.
Thank you for that. That was a really cool explanation. Go ahead, Brendan, if you have a question.
Yeah, ditto, that was a really awesome talk. Thank you so much for sharing that. One of the things I really admire about your project is how much effort you folks have put into making everything interpretable and transparent and open. And I was wondering if you could kind of expand upon that slide a few ago where you said that the hospitals classified a variant as benign, and then Boost DM thought it was oncogenic in a driver. I'm just kind of, can you talk about like kind of what the sentiment has been among your clinical partners regarding like the machine learning model? Because sometimes I think there's hesitancy there.
There's, sorry?
Oh, I think sometimes the clinical partners aren't sure about the machine learning models.
Yeah, sure. Yeah, no, the thing is, so the clinical partners classify it as variant percentage significance and Boost DM as driver. It's not that they classified it as benign and okay, yeah, just this clarification. But so two parts here regarding the second part of the question. Yes, I mean, of course, clinicians are not completely sure about what they should do with this, and I would say we have seen different experience depending on which hospital, which kind of patients. For example, for groups addressing really clinical routine, this is very far from variance percentage significance and being able to apply something that is a prediction to something to the clinical routine. This we see them a bit not super open, but we will try to talk about this and convince them. But on the other hand, people, so medical collaborators that are more related to clinical trials or something more experimental, in this case, they are already actually. Some collaborators were already using CGI before the project as not as the only reference, of course, but as one reference. And in this case, in groups and in clinical context, more patients with no more treatment options, something like this. In this case, we see that they are more open to this kind of predictions, let's say. And regarding in general what we are working it, I mean, this is something that we are starting right now, but what we have seen already is quite several examples of mutations, missense, in a lot of times in tumor suppressors like TP53 or ATM, for example, also some others, that there's not enough evidence to classify them as pathogenic, but we see that there are quite some convincing evidence that they could really be oncogenic. For example, in the specific example I showed you, that is in an important domain, it's in a cluster of mutations observed in patients, it's in a conserved residue. So yeah, this is something that we want indeed to address in the future.
Well, thank you so much for sharing. That's really interesting.
Thank you, Brendan.
Yeah, I think that's really cool. Just a couple comments. I think that, you know, as computational models evolve and become, you know, more sophisticated, it's important that we in this community understand kind of what criteria are being used there, so we can kind of advise the field about the importance of computational models and what exactly we should be looking for when we're using these for variant interpretation in this way. So I think that's a really good background, and if you haven't already, it would make a really great abstract or paper to compare something like Boost DM to like a Rebel score or ClinPred with variants that do have some functional backing. It would be a really neat project that could help drive some of these guidelines.
Yeah, definitely. I mean, this is something that we haven't done yet systematically, but I mean, we did it for the original paper. We compared with some MAPEs. We also compared with some other computational methods, but we want to do this kind of comparisons, also taking advantage of the data from the hospitals that we have access to. And yeah, but of course it would be very interesting to really do this kind of a study.
Yeah, that's fantastic. We all have a lot that we could learn about all of this criteria that goes behind these computational models. So thank you so much for all of your work and all the work from your team. I see Andy has his hand raised, so go ahead.
Slipped in at the last second. Thanks, Beth. Santi, thanks for the great presentation. Really interesting work. Have you had to keep in mind any regulatory considerations for like IVDR use in Europe? I'm curious if that's something that you're thinking about.
Great point. I mean, yeah, this is something. I mean, is one of the objectives of the project to pass the IVDR regulation for the CGI. So yes, we are taking into account all this part. But yeah, I mean, regarding the use of machine learning in the regulation, this is something that actually they are. Olivia Tort is here, so maybe she knows more about this part. But for example, if I recall correctly, the regulations, even the regulations in the EU, are changing now regarding IVDR and regarding the use of artificial intelligence to regulate the clinical devices and all this. So yeah, we are following this, and we will try to adapt CGI to these requirements so that it can be used as a medical device. Yeah.
Thanks. Thanks. That's helpful.
Thank you.
Wonderful. Well, thank you again so much for your presentation and for all the great work that your group has done. You're really a valuable member of this community, even if it is after hours when we meet most of the time. So thank you so much.
Thank you so much for the invitation. It has been a great pleasure to share this with you, because yeah, as I was mentioning at the beginning, we follow really with interest your meetings. We don't come live to the meetings, but we follow all your discussions and papers. So it has been great to share our project with you.
The pleasure is all ours. So thank you again, and yeah, have a great evening, everybody, and we'll see you all next week.